do subsurface drip irrigation systems influence microbial communities? Do we see changes in microbial communities in those soils, near and far from a dripper? I guess where we were getting at was if you do see changes in the microbial community, you are likely to also see changes in tomato yields. We developed a project in the tomato processing industry, and that industry was having problems with subsurface drip irrigation systems and the variability in yield that was occurring along rows and between rows. We offered to use, more innovative approaches, so DNA approaches to assess what was there. But you can even take the DNA approaches further and look at how what functions are occurring in those systems. So it's very sensitive, and you can interrogate the data in a number of different ways. So you can say, are there potential pathogens in those soils? How abundant are those pathogens in the soils? Are these pathogens related to, the conditions created by the emitters? So if close to the emitter you get, say, a low pH, are these organisms are they more prevalent close to the emitter?
Well, the interesting thing is that we were comparing two different fields. So one field that had been under subsurface drip for 5 years, and another field that had only had its system installed a year ago. And what we found was that the soil chemistry in the newly installed field was much more highly variable, whereas the field that had SDI, subsurface drip irrigation, ongoing for 5 years, the soil chemistry seemed to have settled spatially and didn't vary as much as you moved across the field. So things like pH changed quite a lot in the new field when you were moving from row to row, whereas the pH was pretty much stable across the field in the old drip field. And this soil chemistry variability was also reflected in the bacterial community. So the bacterial community was much more highly variable in the new field than in the old field. Because of this, we can get a clue that the soil bacterial community is linked in some way to the soil chemistry in terms of its variability. And what we found was that one of the major drivers of soil bacterial community diversity was primarily pH, and secondarily we found that the cation exchange capacity also affected the bacterial community structure. In the old field, where pH was a lot more stable, we found that there wasn't one obvious soil chemistry driver that influenced the bacterial community. Instead, we found that it was a combination of things, about 5 or 6 different variables, including sodium levels, soil moisture, potassium levels. But all in all, these variables combined could only really explain a small percentage of bacterial community variability, which means that there's a bit more going on. And I guess that's the exciting thing about this, is that we're still trying to figure out how the system works in the soil.
So a lot of this work is pure discovery. We're actually looking, finding things that have never been quantified or identified in a system before. So it's really frontier science. However, we're trying to develop some understanding of how the community patterns are influenced by their environment. And then we've got to work out what the significance of those patterns are and whether we can actually manage them to improve our plant performance systems. I'm Associate Professor Pauline Neely, and I'm based at AgriBio, and my job is to lead a group who's working in soil microbiology.